Welcome back. We are starting with the chapter 6, viruses and other cellular infectious agent, and this will be the last chapter of the module 1. So in this chapter, we are going to talk about something that is not included in the 3 domains of life. It is not a bacteria, it's not archaea, it's not eukarya. Every day we actually do encounter many of these, and these sometimes cause disease, sometimes they don't. They can infect plant, bacteria, animals, every living organism on the earth, and they are obligate intracellular parasites. That means that outside a living tissue, they do not have any living entity. They do not qualify for living organisms, and that's why they are not included into the domains of life. So we're going to find the terms, and we are looking at the bacteriophages, which are the bacterial viruses, and we're going to talk about many different things as well. So the viruses are actually made up of protein and nucleic acid. It does have a box. That box is called a capsid. Think about that way. And inside that box of protein, you have the nucleic acid, which could be either DNA or RNA, but not both. And the viroids actually are naked RNA. They are much smaller, and they are actually only capable of infecting the plants only. The satellites are only nucleic acid as well. If the viruses are actually infecting a tissue, some satellites or the satellite viruses can actually contribute to the severity of any kind of infection. We are going to see that. On the prions, which are not actually viruses at all, these are some of the infectious proteins which actually fold in a different way and becomes infectious. So we're going to talk about these things. So the first thing that what we are talking about is the major cause of many disease, important in a new source of therapy as well. We are actually engineering the viruses to target tissues which are particularly cancer. This is becoming a therapeutic agent as well. And the new viruses are emerging. There are many being discovered. There are some of the giant viruses which were discovered in the just last couple of years that were completely unknown. And that actually, we always used to define viruses. They are super ultra microscopic. That means you cannot actually observe them with a light microscope. But in the recent times, the discovery of a couple of giant viruses actually now make that these giant viruses, which are the only exception, you can actually use a light microscope to visualize them. So they are important members of the aquatic world, and they move the organic matter from particulate to dissolve as well. And they're important in evolution. They transfer genes between the bacteria and the others. And this is actually very interesting part, that in our DNA, very interestingly, in our DNA, human DNA, there are more than 40% or more signature sequences of retroviruses. These retroviruses are actually RNA viruses, which actually, when infect, they integrate into the genome or the DNA of any other animal. So classic example of that is HIV. If for the cats, it's FELV, feline leukemia virus. These are the retroviruses which actually put their DNA inside, put their RNA converted to the DNA inside the genome of the organism they are actually infecting. And our genes actually do contain a large number of these retroviral sequences, and these are all defective. They actually do not cause anything, but they are just there, and it's kind of a molecular signature of evolution, which we can actually tell that when they were infected. And it's an important model system for the molecular biology, being very simple. It's easy to study, and many cell and molecular biological basic idea research, or the basic research, were actually being done by using the virus as a model system. So the next, what we are looking at, that they can infect all cell types. So the bacterial viruses, now remember that what are the bacterial viruses? Sometimes people get confused with the term. Bacterial viruses mean the viruses which infect the bacteria. These are called the bacteriophages or just phages. There are few archaeal viruses as well. and we are learning more and more about them. There are many which are eukaryotic viruses. They infect plants, animal, protists, fungi, all throughout the eukarya. And they can be classified depending upon the genome structure, life cycle, morphology, or genetic relatedness. We're going to talk about these things in a second. So let's see that in this area, 6.2, virion structure, we are going to define by capsid symmetry and presence or absence of an envelope, and we're going to define that what we mean by these. So first, let's take a look at the structure of the virus. The virions are tiny. Now, approximately 20 nanometer in diameter, but I'm not very much fan of this word right here because, yeah, there are some of the smaller ones in the 20 nanometer diameter, but there are routinely viruses which are 40, 50, 70, 100 nanometer, up to 200 to 300 nanometers. So they vary in size, but it is really not you cannot average or tell that. The most viruses must be viewed under the electron microscope, as we said. All of them contain a nucleocapsid, which is composed of either nucleic acid, right here, either DNA or RNA. And on top of that, you actually do have a protein coat, which is the capsid. Now, some viruses consist only the nucleocapsid, that means the nucleus and the capsid, but other actually also have an envelope on top of that nucleocapsid. So these are the enveloped virus, which does have a membrane outside it, right here, whereas these are non-enveloped virus. We often call this as a naked virus, whereas we call this as an enveloped virus. So what are these envelopes? The envelopes are usually membranes. These are phospholipid membranes, which actually the viruses derive either from the plasma membrane or the Golgi or endoplasmic reticulum or nucleus membrane, any kind of membrane which is a phospholipid bilayer. And there are many different types of these spike proteins. Mainly these are glycoproteins that actually are inserted onto that membrane by the virions. And this is a typical structure of these. So let's take a look at the size and morphology of the select viruses. That's why I was not a fan of this 20 nanometer average size, because they are many different size. These vaccinia virus could be 200, 300 nanometer in diameter. Herpes virus is also a big virus. This is a rhabdovirus. It's a bullet-shaped virus. A rabies virus is a classic example of that. These are the T-even coliphage. This is actually a phage which actually infect the bacteria. As we said, a very complex structure. What we mean by the T, we are going to talk about this T number, the T-even and the T-odd. And then you can see that this is a flexuous-tailed phage as well. A very small virus, 5X174. This is actually a sulfobus-shaped virus right here. Human papillomavirus, pretty small. On the other hand, you can see right here, the first virus which is discovered is a tobacco mosaic virus, which is elongated rod-shaped virus. This is a giant virus, which is a mimivirus. It actually was isolated from an amoeba, and this is the first virus which actually can be observed under a light microscope. This could be almost 0.5 micron in diameter, and it is huge. It's large, and it can be, you can see that this is actually approximately 1 micron, and you can see that this could be almost 0.5 micron in diameter, and you can visualize them under the microscope. And this is an acidonas. This is a two-tailed virus, and this is also another giant virus. So these are some of the recent discoveries that you can actually see. So the next thing that what we are looking at, capsid is actually the protein coat, which actually cover the nucleic acid, which could be either DNA or RNA. And the large macromolecular structure, which serves as a protein coat, protect the genetic material made up of subunits. And remember that this is an important word. The capsid is made up of these protomers. And these protomers actually form, which is called a capsomer. Now, I'm just throwing a couple of different words. What we mean by that a capsid, the whole big structure, that can be made up of capsomers. These are some of the unit, but a single capsomer can be made up of several different types of protomers. And I'm going to show this in a different slide with a picture that what is the difference between a capsid, capsomer, and a protomer. And the capsids could be helical, icosahedral, or complex. So let's take a look at the different types of viruses which have different types of capsids. So first one is a helical capsid. So what we mean by that, these are the shaped hollow tubes with the protein walls, and the protomers actually self-assemble. And the length of the capsid is a function of the nucleic acid. So essentially, what we are looking here, this is a typical structure of, let's say, tobacco mosaic virus. It's a rod-shaped structure that you can see. These are all pseudo-colored pictures. So here in the tobacco mosaic virus, this is the RNA, and you can see that this is only just one portion, and right here, this is the kind of a larger portion of that. So in the middle, you have this helical RNA, which is covered within this helical capsid. And what kind of symmetry they have, this helical capsid actually have a corkscrew-like symmetry. It's a screw axis which goes right here. And you can see that it actually is assembled in this way. So each of these units that you are seeing, right, each of this unit, 1, 2, 3, 4, that you're seeing numerous of them, each one of them is a protomer. And these protomer are actually making the capsid. So what happens that when these are actually replicating inside a living cell, that's the only where the viruses can replicate, when these molecules will be produced by the protein synthesis, they will actually assemble around that RNA, will encapsulate the RNA, and they will, the word I used is encapsulate the RNA. What we mean by the encapsulation means that it is wrapping around the nucleic acid to form this helical capsid, and they can self-assemble. On the other hand, here you can see that there is a different type of virus right here, and this is actually an influenza virus right here. And the influenza virus actually do have an helical symmetry. It doesn't have to be a long structure. It could be a just round filamentous, many different types, but they are all helical symmetry. And inside that, they have 8 segments of RNA. These are single-stranded RNAs, and they are right here inside. They're covered in a nucleocapsid, all these RNAs, and then they are packed inside a structure, and this whole structure is covered by an envelope. So this is an enveloped virus with the nucleocapsid inside. This is a naked virus where the RNA is just covered just by the capsid. So this is just the nucleocapsid. This is nucleocapsid inside that envelope. So 2 different types that we are seeing. So the next one we are looking at is the icosahedral capsid. So an icosahedron is a regular polyhedron. It's a polyhedron like this with 20 equilateral faces and have 12 vertices. So you can see that right here, these are the vertices, and from that, you actually get that symmetry. So if we just look at that, the icosahedral symmetry can have a 2-fold, a 3-fold, and a 5-fold symmetry. So what do we mean by a 2-fold, 5-fold symmetry? So think about that right here. If I just cut the virus in half, this half will be one symmetry and this half will be symmetrical. So these are right across from here. If I just cut the virus in half, you will have a 2-fold symmetry. On the other hand, if you just look at these viruses right here, when you have these vertices right here, this right here is 1, 2, and 3. So you can see that you can have a symmetry which is 3-fold symmetry. On the other hand, there are some spaces where you can have 1, 2, 3, 4, 5. You can have a 5-fold symmetry. So let's take a look at what we mean by this type of symmetries. Icosahedral virus can be made up of something which is called a pentamer. And these pentamer is a pentamers or pentons have 5 subunit capsomers. So you can easily see that what we mean by this. So these type of capsomer are made up of 5 or 6 protomers, and this is a 5 subunit capsomer. So let's take a look at when we are looking at the pentons. So if you're looking at the pentons, if you just carefully look at this structure, you have 1, 2, 3, 4, and 5. Each individual these cyan balls that you are seeing, each of them is a protomer. Each one of them is a protomer, and each of these protomer actually form a penton subunit. It's called a pentamer. And here you can see that they could be very, very complex, a typical icosahedral virus. Here is also another thing, that if you just look at these vertices right here, it could be 1, 2, 3, 4, 5 proteins around with that. So you can see that they are sitting on a pentamer axis. But if you can see, like right here, if you're just looking farther around that, you will see that you have 1, 2, 3, 4, 5, 6 structures around that. So you will have the axis is actually different. So pentamer to hexamer structures. So that's all about the symmetry. The next part we are looking at is the complex symmetry, where we have some viruses actually do not fit into the helical or icosahedral capsid. Classic example are pox viruses, which are very large animal viruses. But remember that we are excluding the giant viruses, the mimi viruses, mama viruses. There are many different types of viruses which are much, much larger as well. And there are large bacteriophages actually do have what is called an binal symmetry. And the head actually is icosahedral, whereas the tail is helical. But if we just look at a typical structure, think about that. This is a typical structure of a pox virus, which could be up 250 to 300 nanometer in length, about 200 nanometer in width. And you right here, you have a core membrane, and inside that you have the nucleic acid. On the other hand, the outside of that, you actually have this lateral body, and then you actually have an outer membrane, outer envelope. This is a very, very large virus with many, many genes that actually is coded by that. So you can see that this actually doesn't fall into the helical symmetry. This does not fall because it is all different type of shapes. It is not a fixed type of shape, and that's why you cannot actually tell it's a helical symmetry, because there is no corkscrew axis that you can actually see what is found in the helical symmetry. So that's why it is a very complex structure. So next one, which is also a very complex structure, is the bacteriophage, and you can see that these are called a binal symmetry because the head actually is icosahedral with this nucleic acid inside. So you can easily see that this is, of course, icosahedral, and these are the equilateral triangular faces. And a typical one has 20 triangular faces that you can see. And then right here, you have a collar, you have a sheath, you have a base plate, then you have these short tail fibers, tail pins. All of these are very, very complex structure. So overall, if we're just looking at bacteriophage, here is an electron micrograph, and there is a cartoon. So how they actually infect the bacteria. So these bacteriophages actually sit onto the bacteria or engage them onto this on their cell wall by these long tail fibers. So they absorb or attach onto the bacterial surface through this. Then what happens, that this sheath right here is actually ATP driven. ATPs are actually loaded into that. It actually lowers that sheath, and it actually pushes or thrusts these tail pins inside the bacterial cell wall, say peptidoglycan layer. And then what happens, there is an ATP driven motor right here. It will actually bore a hole through the peptidoglycan layer, breaching the peptidoglycan and the plasma membrane. So this actually will rotate. Once that hole is made, then the capsid head will actually squeeze that nucleic acid, and it will pass through this collar and the sheath, and that will be going into the cell. And this is a classic way that how the cell wall of the tough peptidoglycan layer of the cell wall of the bacteria is breached by this process. Now, this is not needed by the animal viruses because they just only have the plasma membrane, and you do not have to bore through the tough peptidoglycan layer to deliver the virus inside the cell. It is much easier for the animal viruses to infect an animal cell. So next, what we are looking at is the viral envelopes and the enzymes, and many viruses are bound by an outer flexible membranous layer, which we call as an envelope. So here these are all pseudo-colored pictures. So here you can see that this is a bullet-shaped virus. A typical rabies virus is like this. It is a family of Rhabdoviridae, rabies virus, bullet-shaped virus. So right here, you can see that these red structures that you are actually seeing, these red structures are the membrane glycoproteins. This dark structure that you are seeing, that is actually the envelope, which is a membrane. And these glycoproteins, which are sticking out, it actually anchored onto that membrane. And inside that, you have the nucleocapsid, which is the RNA. It's an RNA virus with RNA covered by a protein. So this is a typical structure of the viral envelope. Here you are seeing that this is also a large virus, which is a herpes virus. And inside that you actually have a this is a nucleocapsid. Outside the nucleocapsid, also you have quite a bit of a space. And this space is also called tegument right here. So this is made up of a tegument protein. And then you have an envelope. And this envelope is also, again, derived from some kind of membrane, which from the animal cell. And we're going to talk about that, where they can derive it. They can derive it from the plasma membrane, endoplasmic reticulum, and the envelope, which is actually derived for the herpes virus particularly. You do not have to remember this, but they actually derive it from the endoplasmic reticulum and Golgi complex. So the glycoprotein actually have these beta envelope spike proteins, which are inserted into that membrane as well. So what you are seeing, that animal virus envelopes with a lot of these lipid carbohydrates usually arise from many of these different types of membranes. But again, it can vary. Plasma nuclear membrane, that is written here. But remember that many of them arise from the endoplasmic reticular membrane or Golgi membrane as well. Any kind of endomembrane complex. So there are many viral envelope proteins. These envelope proteins, which are virally encoded, may project from the envelope surface as spikes, or which is called a peplomer, involved for the attachment mainly. And these are also used for the identification of the viruses. What we mean by that, a classic example I'm giving, we all talk about this influenza virus, and we talk about this H1N1, H5N1, H2N2, and we use these terms constantly. So the H actually stands for hemagglutinin, and the N stands for a neuraminidase. These are the two glycoproteins that are present on the human influenza viruses. And depending upon the combination of these two proteins, which is HA and NA, or H and N, hemagglutinin and neuraminidase, we actually classify the viruses in different groups as well. And they may have enzymatic and other activity. Say neuraminidase in the influenza virus is actually really required for actually not for infection, but to release the virus from the cell so that they can pinch off from the plasma membrane to go out into the environment or outside the cell. We're going to talk about that sometimes later. And also may play a role in nucleic acid replication as well. And we're going to talk about that. So the viral genomes are structurally diverse. A virus may have a single-stranded or double-stranded DNA or RNA. So remember that. So the DNA could be double-stranded, or it could be just single-stranded, one strand. RNA could be double-stranded, and it also could be single-stranded. And the length of the nucleic acid also varies from virus to virus. Genomes can be either linear or it could be circular. So some RNA viruses have the segmented genome that we have seen. So the next one that what we are seeing, we are looking at the viral life cycle. So the first thing that what was done, that in the early experiments in 1939, Max Delbruck and Ellis, they actually was working with a bacteriophage T4. Now here, see, T4 is a T-even bacteriophage. Remember that if it is a T3 or a T5, it will be a T-odd bacteriophage. And this T number is called the triangulation number. And this triangulation number has something to do with the icosahedral symmetry. So from this T number, we can actually tell that the virus will have how many triangular faces. So that's the T number. So here what we are seeing, that when we actually infect the phage in culture, which is going to infect the E. coli, after the infection, you actually have a period which we call as an eclipse period. You actually don't see any viruses whatsoever. There is no viral growth or anything like that. So what it is doing in here? So after that, suddenly you can see that there is some viruses coming out, and then there is a burst that you can actually see right here. So this is basically the burst, means that at the initial phase, which we call as a latent phase, there is absolutely nothing. And then suddenly there is a burst of viruses which actually comes out. So what happens in this period, the eclipse period or the latent period? At this point in the eclipse period, the virus inside the cell actually is doing a lot of different things. It took control of the cell and it is replicating its genome and making all the proteins, which makes the protomer, capsomer, and the capsids to enwrap around that protein to make another many of these virus particles. So that's what the virus is doing inside the cell and in the eclipse period. Once these are already assembled, all these viruses are made, they will actually come out of the cell with a burst. So these are actually some of the first step and evidence to understanding the viral life cycle process that was done by this process, that how they actually do replicate. So right here, we are looking at the viral multiplication, and the mechanism depends upon the viral structure and the genome, and it could be very complex. But the steps are similar to some extent. They first attach to the host cell, you have the attachment, then they will actually enter the host cell, and they will actually uncoat the genome because its genome need to be delivered inside the cell. Then once the genome of the virus goes inside the cell, that genome is somehow takes control of the cell. The cell will actually stop doing its own function or making its own protein. It will make some of them to some extent, but it will mainly engage in actually replicating the viral proteins. And that's what it is actually the most important part, that it will synthesize the viral proteins. It will actually transcribe the viral genome into the mRNAs. It will make its own genome as well. And after the mRNA, it will actually make the viral proteins, and then you will have assembled. So the genomes that is being made, it will be assembled into the virion particles. The infectious particles of the viruses are called the virion. They will be assembled into the virions, which will be released from the cell. And there are two ways that the virions are released from the cell. Either they will actually lyse the cell, the cell will burst and come out, or it allows the cell to live for a little while. They will actually bud out or bleb off the cell, and they will actually in that process steal a portion of the membrane while budding out. So either budding or lysing the cell. So let's take a look at the attachment, or the first part, which we called as an adsorption. And there are specific receptor is required for this attachment. And the scientists always look for these receptors so that they can actually design the drugs to do something. And the receptor actually determines the host preference of the viruses. There may be specific tissue tropism. That means, see that the specific, say for influenza virus or the rhinovirus, which causes the common cold, they will have specificity only for the lung epithelial tissue or lower respiratory tract epithelial tissue. They do not have any affinity for any other tissues in the area. And it will be maybe more than one host. There are some diseases which we can actually see in other animals, so their host may actually vary. On the other hand, they actually may involve in more than one receptor. Say, for an example, the human influenza virus, water birds or waterfowls are actually the reservoir for that. So these waterfowls actually one of the reservoir. On the other hand, see the pigs or the swine, they are also one of the incubation agent for the influenza virus. So you can see that the influenza virus actually can get into the pigs. It is actually the reservoir original is actually birds. So more than one host. And why I said that these different types of animals, like the birds and the birds as well as the pigs or the swine, they actually do have preferences for different receptors in there as well. So so they could be very specific depending upon the different receptors. So what we mean by that one virus could be very virulent to one strain to the human beings, but it not be that much virulent to the pigs because it actually contain a different type of receptor, and that may allow a different viral strain to infect that, but not the virulent strain which is very virulent in, say, human beings. So these type of variations can actually happen. So the next what we are looking at is how the virus actually enters the cell. Either the entire genome or the nucleocapsid actually gets into the cell, and this technique actually, or the mechanism actually, varies between the naked or the enveloped viruses. And there are three methods: either the fusion of the viral envelope with the host membrane, endocytosis of the vesicle, or the injection of the nucleic acid. So let's take a look at that, what we are looking at in this picture. Right here in the first set of picture, we are looking at that the entry of the enveloped virus by fusing with the plasma membrane. So this is the plasma membrane with the receptors, and this is a typical enveloped virus. So you have an envelope which is blue in color, and then you have these tiny small spikes which are usually the glycoproteins. And when the virus actually just accidentally bumps onto a cell surface which have the specific receptor for these specific glycoproteins, which are these spike proteins, they will actually lock into space. So when the viral proteins and the receptor will lock in space, remember that the receptor for on the cell surface is supposed to do a different function, but it's just the virus exploit that. Say, for an example, giving a classic example, that LDL receptor is a low density lipoprotein receptors is exploited by many viruses to bind and get into the cell. So once the receptor binds to the virus, it will actually fuse with the membrane, and then what happens after the membrane fusion, the inner nucleocapsid is actually released into the cytoplasm. And once that is released into the cytoplasm, the genome from here will extrude out, and it will travel to the nucleus to do some difference. If it is an RNA virus, it can actually be utilized to do different types of work in the cytoplasm as well. In that case, the virus actually carries its own enzymes to take care of all the different types of events that's going to happen. So this is a typical mechanism by which the membrane fusion happens. There is another process, which is called the enveloped viruses get inside by a process of endocytosis. So what is the difference between here? That you can see that there are receptors. When the virus binds to the receptor, it actually makes a dip right here. This dip actually gets deeper and then finally pinched off. This is an endocytic vesicle, and you can see that the endocytic vesicle is actually coated by some kind of protein. And usually these are called clathrin, and these are clathrin-coated vesicles. So what happens that although the endocytosis is supposed to get the cell some important material like a food material or something like that, but this receptor-mediated endocytosis actually got something which is actually detrimental to the cell. So once this endocytic vesicle is actually pinched off inside, on the membrane of this endocytic vesicle, there are actually proton pumps. They will actually pump protons inside this endocytic vesicle. So what we mean by that, in the cytoplasm, you have lots of these H+ ions, or the protons. The protons will be pumped inside the endocytic vesicle. So once these protons are pumped into the endocytic vesicle, the acidity inside that endocytic vesicle actually rise, and that will cause this membrane, or the viral membrane of the enveloped virus, to fuse with the endocytic membrane. So this is the endocytic membrane. High pH, or the pumping of the H+ will cause these two membranes, the viral membrane and the endocytic vesicle, endocytic membrane, to fuse. Once that fusion actually happens, it will actually release nucleocapsid right here inside the cytoplasm, which will usually carried by microtubules, by motor proteins towards the nucleus, where it delivers its nucleic acid into the nucleus usually. On the other hand, right here, what you are seeing, that these are the non-enveloped viruses doing the endocytosis, where you do not have a membrane. It is the same process, but here the pH is not pH mediated, because what happens that this virus actually have a protein coat, not an envelope. The envelope can be actually activated to fuse with the endocytic membrane by the pH, but here you do not have an envelope protein coat around that. So in this process, there is a different mechanism by which different types of viral proteins actually inject the nucleic acid inside the cell, and the protein coat actually remains in the endocytic vesicle. So these are some of the mechanisms by which the cells actually can receive the viral nucleic acid. So here is a synthesis stage. What we are seeing that these are some of the ultrastructural micrograph of some of the vesicles, and these are exploited in replication and the transcription process. So what happens, the genome of the virus actually dictates that what's going to happen in the cell. And the double-stranded DNA is a typical flow. In the RNA viruses, it must carry in or synthesize the proteins necessary to complete the synthesis. What do we mean by that? Say, for example, if it is an RNA virus, remember that there is no mechanism in the eukaryotic cell to replicate RNA from RNA. Again, repeating, in an eukaryotic cell, you cannot make RNA from RNA. RNA is always made from the DNA to RNA, and that's done by the RNA polymerases in the eukaryotic cell. There is nothing called an RNA to RNA polymerase, but the viruses actually having RNA genome do carry an enzyme, again, remember that, do carry in the enzyme, which is called RNA-dependent RNA polymerase, or RDRP. RNA-dependent RNA polymerase is the viral enzyme, and there are many, many different types across different genera of the viruses, which actually will do both replicate and transcribe from the viral RNA. What do we mean by the replication transcription? Both the RNA is converted into an mRNA, that is your transcription for making the protein, and then RNA genome is made into an identical copy of an RNA genome, because that is needed to be packaged inside a new nucleocapsid to make the virus. That's a very important concept, that in the replication process, the viral genome, which is, if it is an RNA, need to be replicated to an exact replica copy what came in, making many of those, and then those individuals needed to be put inside a capsid or nucleocapsid so that the virus can actually function. So the genes or proteins may be referred to as early, middle, or late. There are some genes which are transcribed early. They make mRNAs, which actually regulate some of the genes which will be expressed in a middle portion or late portion of the infection cycle. And it's a complicated process. And sometimes what happens, they may induce the formation of these membrane-protected replication complexes as well. Here you are seeing that you have some membranes of the endoplasmic reticulum, and along with that, there are many of these vesicles. A typical classic of these vesicles, which has a bilayer. So you can see that you have an inner purple layer. This is a lipid bilayer, and outside that, you have a lipid bilayer. So remember that it is not a single membrane, it's a double membrane viral vesicles. So two lipid bilayer on the outside, two lipid bilayer on the inside. And the viruses actually replicate inside. A classic example, many viruses does that. Classic example, poliovirus does this membrane-bound replication as well. So the next one, what we are looking at is the assembly, that this is actually done by like a conveyor belt, like a assembly plant in a, say, car manufacturing company. One at a time, this is first the base plate proteins come in. Base plate is formed. Then the tube is attached and formed. Tube and sheath is coming together. And on the other hand, see that the head proteins making a prohead, then the mature head with the DNA, then whiskers on the and the neck actually come in. Then they put the collar in, and then long tail fiber proteins actually attach onto the base plate, and that is the process. And these late proteins are important for the assembly, because think about that. This is why it is late protein. Initially, when a virus doesn't need to be this bacteriophage, it could be any viruses, the nucleic acid comes out, and the first thing it will do is to transcribe into mRNA, and that mRNA will produce these proteins. That's the first thing. So making of the proteins which will actually make these structures and the different functions, those will be the early genes. And in the middle portion, they will make more of these proteins or structural proteins. And in the late phase of the infection, the proteins are actually important for the assembly. So what will happen that in this process is actually driven by some of the proteins which actually will make this assembly. And why these late proteins are important in this assembly? These late proteins actually are checkpoint proteins. So each time a structure is modified into a structure as being made, these late proteins actually check whether this structure is correct or not. If it is not, it will go back to the previous step and it will be corrected. And that's a very, very complex assembly process. You can easily see that. So the base plate, tail fibers, and the head components of the bacteriophages are actually completely separated assembly, all coming together just like an assembly line in a factory. So next, what happens that after doing this, the non-enveloped virus actually lyes the host cell. And right here, you can see that this is a sulfobus bacteria. And these viruses are actually coming out by lysing the cell. You can see that this is right here. This is the head, and this is the sheath or the neck and the sheath of the bacteriophage. They were all inside the cell. And after they fill up the cell, they're lysing the cell. So here is the plasma membrane and the cell wall. And you can see that this area is ruptured, and all these virions, which are the bacteriophages, some kind of, are coming out of the cell. This is called the cellular lysis, which is done by the non-enveloped viruses. We are going to see that there is a different mechanism for the enveloped viruses. Enveloped virus use, on the other hand, the process which is called the budding. So in this process, what happens that the virus actually take all these different proteins and then transport these proteins by the secretory pathway through the Golgi, and these proteins actually reach the membrane. And again, this is a typical where it has been shown that this is a plasma membrane. But remember that instead of plasma membrane, this could be a nuclear membrane, this could be an endoplasmic reticular membrane, or the Golgi membrane. Each virus actually do have a specific area from where it actually buds. It doesn't have to be plasma membrane. But once these glycoproteins or the membrane proteins are synthesized and transported right here, and there is a very interesting transport machinery. Many of these are actually secreted through the Golgi, and sometimes they are actually transported by motor proteins from the inside of the cytoplasm all the way into the plasma membrane of the cell. Now, once they are transported in here, the nucleic acid actually comes in, and this is the nucleocapsid, which is basically the nucleic acid wrapped with the RNA, wrapped with the protein. That's the nucleocapsid. When the nucleocapsid comes in close, the interaction actually changes the dynamics of this area, and that actually triggers a kind of formation of this budding virion. So once this virion actually buds off, it is still attached to the membrane. And here this is written as a neuraminidase because this is giving an example of just say typical influenza virus. These molecules are actually sticky. These receptors right here, there are some receptors, remember that what I'm saying, that there are some receptors which are going to be attached onto a membrane of another cell. So what happens that at the initial stage, these receptors actually attach themselves to the proteins on the plasma membrane from where they are coming in. So you need this neuraminidase which actually cleave off, and it allows the virus to escape so that they are not attached anymore in there. So this is the neuraminidase is used to pinch off and bud the virus out of the plasma membrane. So sometimes the viruses actually use the host actin tails to propel through the plasma membrane. This is actually used by the pox viruses. What do we mean by that? The virus is actually right here, and then the actin tail actually forms underneath it, and it pushes the virus outside. It pushes the virus outside and makes a bleb, and from there, the virus is actually released. Classic example in pox viruses. So next, we are looking at that the bacterial and the archaeal viral infection. There are many different types. The virulent phage, which are the bacterial viruses, have one reproductive choice. They multiply immediately upon entry. They lyse the bacterial host cells. And on the other hand, the temperate phage have two reproductive options. They either reproduce lytically, as virulent phages do, or remain within the host without even destroying it. And many temperate phages actually integrate their genome into the host genome became what is called a prophage or a lysogenic bacterium. And in the relationship is called a lysogeny. And this is the same way that the retroviruses actually infect us and establish themselves or take their RNA, convert it to the DNA, and that DNA is integrated into our gene and it becomes a provirus. So the retroviruses become a provirus when it's infecting an animal. And this is the same thing, that this is a prophage, is the integrating phage into the chromosome of the bacteria or the archaea. What they do, they can have virulent genes. They can actually change or alter the bacterial outcome, or what the bacteria will actually do, their infectious pattern. But one fine time, and it is not known, that at one point of time, these temperate phages can actually excise their DNA out of these bacteria or the archaea, and they can go into the virulent phage form as well. So this is actually one of the very interesting area of study. So let's take a look at the lytic and the lysogenic cycle in the picture. So right here, bacterial cell, which is a large bacterial chromosome, this is a phage DNA. And after infections, what you are looking at, that this phage DNA is injected, immediately that DNA will destroy the bacterial DNA, and it will utilize all the bacterial enzymes to replicate its own DNA, the phage DNA. Once it's along with the replication, it will also transcribe the mRNAs from the genes of these host cell from the prophage DNA. So once these mRNA from this prophage DNA are made, they will actually make all these components of the virion. Remember that you have some early genes, there are some late genes, then there are some middle genes. So most of the time, the early mRNAs or the early proteins will actually control the cellular metabolism to stop everything and actually help the virus to make its components in that state. And the late genes will actually make proteins which actually help in assembling these particles. So what you can see, that these are the tiny phages which is produced from the mRNA, from that the translation of the proteins which actually was made inside the cell. And the virus, actually the word that we use, that put that nucleic acid inside that protein capsid, and that process is called the packaging of the genome. And this is the same word that, think about that, we are packaging something inside a box. So you take a box, if you just pack something inside, packaging. So think about that the bacterial capsid is a box, and you package the genome, which is the DNA or RNA. Here it is DNA, is packaged inside. Now these viruses are making millions and hundreds of thousands of these bacteriophages. Here is bacteria. It will lyse the cell, and they will come out, infect new cells. On the other hand, something can happen, that the phage DNA can actually get integrated right here into the bacterial genome as well. So this is right here, the bacterial genome is inside. Now what happened when the bacteria is actually dividing, all of these bacteria are the lysogenic bacteria. What it means by that in their chromosome, a genome of a phage is already integrated. So once the genome is integrated, all the cells that will be divided or dividing from here, all the cells will have that lysogenic or will be the lysogenic bacteria. What do we mean by that? That all of them will have the sequence of the phage. Now, one fine morning, and without anything which you call as an induction, these viruses actually can excise. This is really not a virus. We call this a provirus, or the sequence of the DNA. They can excise themselves out from this, and they will start actually making mRNA, early, middle, and the late proteins out of those, and they will assemble into this process. So essentially what happens, that many of these exposure to stress, such as UV light, triggers this process, which is the excitation from the host. And when this excitation from the host actually happen, it can just go into the lytic cycle. So this is basically the lysogenic cycle being converted into the lytic cycle. The next thing what we are looking at, lysogenic conversion, means the temperate phages changes the phenotypic type of its host that we were talking about. Bacteria become immune to superinfection. Phages may express pathogenic toxin or enzyme. On the other hand, the two advantages of the lysogeny of the viruses, the phage actually remain viable but may not replicate, and the multiplicity of the infection ensures the survival of the host cell. So what we mean by the multiplicity of infection, or MOI, that there are many of these cells which are lysogenic. Some of the viruses can actually go into a lytic cycle, which will actually continue the infection, but many of the bacterial cell will actually just not do anything. So this actually allows the phage to survive, and because if it is killing all the, this is a beauty of the nature, that if the phage is virulent all the time and killing all the viruses, soon there may not be enough viruses and the replication of that particular phage will be troubled because there is no host. All the hosts are dead. There is no one to infect. On the other hand, if it's a lysogenic condition goes on, then there will be a balance in between the viral replication and the amount of bacteria that is available for new viruses to infect. So this balance is maintained by these lysogenic or the temperate phages. And under appropriate conditions, the infected bacteria will lyse and release the phage particles, and occurs when the condition of the cell causes the prophage to initiate the synthesis of new phage particles, and this process that we call as the induction. So archaeal viruses may be virulent or temperate, just like the bacterial viruses as well, and may establish chronic infections. And really, this is an area that the scientists are trying to understand, and very little is known about the mechanism and how they regulate their replicative cycles as well. So on the other hand, in the eukaryotic infection, there are different types of infection that can happen. So the cytocidal infection results in cell death through the lysis. And the persistent infection may last for years. So these are the different types. In one type of infection, you can see that in the acute infection, it actually kills the cell immediately. So the cell is dead. On the other hand, the persistent infection could be two different types. You can have a latent infection. What we mean by the latent infection, that the virus is inside the cell or not doing anything. And a classic example of that we are going to see is the chickenpox. So the chickenpox is caused by a herpes virus, or think about a cold sore virus. So these viruses are actually present in our skin cells, or for say chickenpox, which is by the varicella zoster virus, and varicella zoster virus is also a herpes virus. They reside themselves into the neurons of the peripheral nervous system, which are called the sensory neurons. They lodge themselves into the sensory neuron, and they do not do anything. They just do cause a latent infection, not causing anything. One fine day, due to some stress or some triggers, these viruses can come out from those sensory neurons and start replicate, and that causes shingles. So acute infection is chickenpox, and that becomes latent into the latent and a persistent infection, which will have lifetime. So no matter what, if someone actually have chickenpox as a young, they will have the varicella zoster virus in their sensory neuron. And that's the reason that the doctors actually tell that after 50 years of age, you should actually get a vaccination for the shingles, because it's a very, very painful disease. On the other hand, a chronic infection is where the virus cannot be cleared by the immune system of the host cell there forever. A classic example of this kind of chronic infection is your hepatitis infection. Hepatitis C virus infection is a chronic infection, and that can cause degenerative disease abnormalities like liver cirrhosis, or sometimes liver cancer even. So the last one, as we said, cancer, that the viruses also can actually transform cells. And what we mean by the transforming cells, that they can become malignant cells. So there are at least more than 20 types of viruses, which we call as an oncogenic virus. That means they are actually cancer-causing virus. How they do that? Either these viruses will actually activate a proto-oncogene. So these are the genes which are suppressed, not doing anything. They will activate a proto-oncogene, and that proto-oncogene will become an oncogene that will actually make the cell cancerous cell or a malignant cell. Or there are many genes which are actually called the tumor suppressor genes. Tumor suppressor genes are specifically in the cell to check uncontrolled cell division. So what the virus actually does, they actually shut down the function of the tumor suppressor gene. So what happens, the cell actually now goes into uncontrolled cell division. So this is how the viruses can actually transform cells into become the malignant cell. Classic example is the human papillomavirus can cause the cervical cancer. So the tumor is a growth of lump of tissue, and benign tumors are actually remain in place. And the benign tumors are actually not cancerous. This could be a benign tumor could be just like a skin tag. On the other hand, the tumor is a growth or lump of tissue, but the tumor could be benign, but also it could be neoplastic growth. So the neoplasia is an abnormal new cell growth and reproduction due to the loss of regulation. And the neoplasia or the neoplasticity will actually cause the cancer, and that will cause some other issues. So let's take a look at some of the definitions. Anaplasia is the reversion of a poor, primitive, or less differentiated state. So what we mean by that, from one particular organ, due to this neoplastic growth, new cell growth and reproduction, if these cells growing becomes a different type of cell, say for an example, if a neoplasticity was observed into the liver, and in the liver, instead of making more cells which are hepatocytes, it's making a kind of a cell which are more primitive and does not have anything to do with any kind of liver cell. It's an entirely different kinds of cell. That is called anaplasia. And when there is a neoplasticity or neoplasia, which is the cancerous tumor, that also metastasizes. That means they spread the cancerous cells throughout the body. It travels from one area to other to cause the cancer. Benign tumors remain in place. They do not metastasize, but the neoplastic tumors will actually metastasize. So the carcinogenesis is a complex multi-step process and often actually involve oncogene, which these are the cancer-causing gene, and they actually may come from the virus, or they may be transformed host proto-oncogenes involved in the normal regulation of the cell growth and the differentiation. And we are not going to the detail, but this is just an overview of that, how the virus can actually induce cancer. There are many of them as well. So the possible mechanism is that the viral proteins bind to the host cell tumor suppressor proteins, and they carry oncogene into the cell and insert it onto the host genome. And a classic example of that, say for example, this tumor suppressor gene, just not going into the detail, just an overview of that. There is a protein which is called retinoblastoma protein. This retinoblastoma protein actually cause, if it is non-functional, it can cause eye tumor in children. So what happens that certain viruses actually bind to this retinoblastoma protein and make it non-functional. So what is the function of the retinoblastoma protein? Retinoblastoma protein actually do control the cell cycle regulation. So cell cycle will not go into the cell division due to the retinoblastoma protein, which actually recruits some of the proteins which actually doesn't help nucleic acid to relax. And this is done by the histone deacetylase proteins. And the histone deacetylase proteins actually relax the nucleic acid so that they can actually go into the replication cycle. But what happens that the retinoblastoma protein, again, I'm just giving you an example. You do not have to go into the detail, but the retinoblastoma protein, when functional, which will not allow the cells to recruit that histone deacetylases, so that this protein actually doesn't allow a typical cell to grow into the cell division if it is not in the right condition. So what happens that there are some viral genes, say for example, there are proteins called T antigens. This T antigen is a classic protein which is actually present in many different viruses. And this T antigen actually binds to this retinoblastoma protein, and they can actually make it non-functional. And when it is non-functional, that means the cell will go into the uncontrolled cell division, leading to cancer. Some cell will actually carry oncogenes into the cell and insert it into the host genome as well. And how it actually happens that the virus actually got this gene, because sometimes when a virus actually, these are usually done by the retroviruses. So what we mean by this, these retroviruses actually do carry these oncogenes. And when these oncogenes being carried by the retroviruses, they actually insert themselves into the genome of an animal. These viral oncogenes will actually become active as the virus is, as the genome is being replicated, and that will cause cancer. They will alter cell regulation, and also they called insertion of the promoter or enhancer next to a cellular oncogene also cause this. So what happens, this is also an interesting idea, that suppose you have a cellular oncogene, which is a proto-oncogene, and this actually do have its own promoter or suppressor or enhancer, which actually doesn't allow the oncogene to express. So it is basically silenced by the cellular promoter or enhancer. But what happens that some of the retroviruses actually can bring in a promoter or enhancer and insert the promoter enhancer, which is this is a viral promoter or enhancer, which they can insert upstream of a cellular oncogene. Classic example, I'm giving an example, and if you do not have to remember, that HIV actually do contain some region which is called the LTRs. These are the terminal repeats, long terminal repeats, which are present on the each end of the HIV genome. So suppose an HIV is infecting a human being and it is a retrovirus, so that HIV DNA, after converting from RNA to DNA, which is a retrovirus, that DNA is going to integrate into the host genome. And once it's inserted into the host genome, these LTRs are actually extremely strong promoters and promoters. But it means that anything downstream to the promoter is going to express. And somehow, think about it, that the HIV has a promote LTR at each end, one end, another end. Somehow, if the HIV actually goes into a cell, integrates into the genome, and this LTR promoter actually replaces and sits just next to a cellular oncogene, now this cellular oncogene lost its original promoter, which doesn't allow it to express. Now you have an LTR, very active promoter, and the RNA polymerase of the cell will actually copy or make transcription of the cellular oncogene. Now this cellular oncogene is active, and that will cause the cancer. So there are many complex mechanisms that there is no time for here to discuss, but these are some of the processes. So here we are looking at that how we can actually cultivate and numerate the viruses. So this actually requires the inoculation of an appropriate host, and hosts for bacterial archaeal viruses are also done routinely and usually cultivated in broth or agar culture, suitable for actively growing bacteria as such. So how you know that a virus is actually growing in a cell? Very, very simple process. Think about that when you actually have a broth of bacteria, suppose, and these bacteria is actually turbid in nature because the bacterial cells go to scatter light, it's turbid. Now you have the virus inoculated into that, and you just wait over the period of time, you are going to see that the turbidity will actually getting clear. It will lose turbidity. Why it is losing turbidity? Because if it is a virulent virus, it's going to burst the cells. So all these bacterial cells are going to burst, and these virions are going to come out of the cell. And that's the reason that they're not going to scatter light, because there is no complete bacterial cell and lose turbidity. And also the plaque assay is a very classic assay that is actually used to enumerate the viruses, which are done by the animal cells as well as can be done by the bacterial cells as well. So the tissue or the cell cultures are infected with the virus phages, and we have studied that in some different context of cell biology. And the viral plaques are localized area of cellular destruction and the lysis that enlarge as the virus actually replicates. So here, think about that this is actually a lawn of E. coli cells. And you actually take some of the viruses and inoculate the virus initially. You just allow these T4 or these phages to absorb onto these E. coli cell for a certain amount of time, say an hour, and then you just rock them back and forth, and you actually take out all these viruses that you inoculated, and then you just wait. After a couple of days, what you're going to see that you will have an E. coli lawn right here like this, but these viruses actually are killing the E. coli and making that area clear on the agar. And each one of these dots is a plaque, and you can count them. And the beauty of this experiment is that each plaque is actually from one virus. You can argue that if there are two viruses close together, of course, but again, one plaque comes from just one virus, so you can actually quantitate the amount of virus from there. And also, if you actually suck up the agar with a pipette tip from one of these plaque, then you actually have a clone of a virus which is very pure, because it's just one virus, it's not a mixture. So the cytopathic effect, on the other hand, is observed into the cell, like mainly the larger cell like animal cell. And in this microscopic or macroscopic degenerative changes or abnormalities in the host cell and the tissues. And also, the host for animal viruses are embryonated eggs. Now, these are actually used to actually make vaccines. So routinely, the influenza viruses are actually made on embryonated eggs. And these are actually done by robots. So I mean, like, if you can just look at how the influenza virus is being made. So tens of thousands of these eggs, which are embryonated eggs, tiny robot actually drills a hole on the top, inoculates them, and actually seals that top, and then the virus actually grows in those eggs. So these are classic example of that, how the animal viruses are actually grown. There are plant tissue culture, and the plant viruses actually grows in plant, and the cultures of separated cells can done. There are plant protoplast cultures, which are also can be cultured plant viruses, and some are suitable for whole plants, and they may cause these localized necrotic lesions and general symptoms of the infection as well. And now the classic example of that you're going to be amazed with that there are many recombinant viruses, which are now initially animal viruses, but you can actually grow them, these recombinant viruses, into the plant. So a recombinant variation of the influenza virus is now grown in the tobacco plants, and that's for the vaccine purpose. So this is not a normal infectious or virulent influenza virus. It's actually a modified or attenuated virus, which means that it's lost its ability to infect a normal host, but it retains all its antigenic properties. And that is recombinant in a way that it can infect and grow in a plant cell. And these are used so you can actually harvest these tobacco plants, recombinant plants, and you can actually extract the viruses from there as well. So to make the vaccines. So here is a quantification of the viruses, and just gives you that how the plaque formation actually happens. And this is a direct counting of the viral particles, and this is an indirect counting is observable by the effect of the viruses. So the different types is an hemagglutination assay. So in the hemagglutination assay is done by a couple of typical viruses. A classic example of the hemagglutination assay is the influenza virus. So the influenza virus actually do contain on its surface, there's a glycoprotein which is called hemagglutinin. So what happens that this hemagglutinin actually cross-link the red blood cells. So if you actually take the blood in a multi-well plate, and if you add the different concentration of the influenza virus, depending upon the concentration, you are going to see that there will be the clumps of red blood cells. So they will actually float around. So on the other hand, if you do not have enough amount of virus, which will not cause hemagglutination, soon all these cells will actually go into the bottom of the well, and you will see a tiny red dot of red blood cells at the bottom of the well. But if you do not see that tiny red dot at the bottom of these wells, that means that they are actually connected to each other by this hemagglutinin, which makes a lattice network of the red blood cells, and that large lattice network is floating into the media. It's not sinking down, and that's the reason you do not see a red eye, which is just a distinct red spot due to the precipitated red blood cells. So that's your hemagglutination assay, and you can just quantify the viruses by that process. Plaque assay, on the other hand, depends upon the dilution of virus, and you prepare a lawn and you actually calculate the number of plaques from a particular dilution, and from there you can actually get the plaque forming units. That means that how many of these viruses actually formed plaque from your culture. So let's see that how this actually happened. So right here you can see that here you have the bacterial cells, or this could be animal cells. So the plaque assay classically can be done in animal cells. You infect the cells and phage is actually killing these bacteria. So these bacteria, once being killed, you can see that more and more bacteria is being killed, and it is actually basically clearing of an area. So you will be able to see this clear plaque, and that you count, as we saw in the previous picture. And in this process, you can actually quantify that how many of them are the plaque forming units. So what we mean by the plaque forming units, why not virion, or how many viruses? Now remember that in your viral mixture, you may have a lot more number of viruses, but many of them are defective. The plaque formation will only happen by only the viruses which are capable of infecting the phage or an animal. So plaque forming unit is from, these are the active virions which are capable of infection, but that is not the number of viral particle. Viral particle to plaque forming unit is actually a ratio, and some viruses like Semmeliki-Forest viruses, almost every virus out of 2 viral particle, every one of them is infectious. So you can see that 50% of the viral particle are infectious. On the other hand, you can see that there are some of the viruses, the plaque forming unit is actually 100 times less compared to the infectious virion particle. Classic example is a papillomavirus. So if you take papillomavirus, out of 10,000 particles, probably just 1 is actually infectious. So you can see that the number of virions does not mean that its infectivity is very high, because most of these are defective. Why this is actually depends upon that they just have a tendency to actually make defective particles. And this actually has something to do with their virulence as well. So this is a measure of virulence. So when we are looking at measuring the biological effects, and we are actually continuing that we just talked about, that determine the smallest amount of virus needed to cause the infectious ID, or the LD50, which is the 50% lethal dose for the host cells or organism. So that's called the ID50 or the LD50. So here you can see that percentage of the cells killed is 100%. And this actually is going down over the period of time. On the x-axis, you can see that this is actually the dilution of the viral particle. So right here, you can see that the viral dilution is about 10 to the power negative 5. At the 10 to the power negative 5, if I just go right here, you can see almost like probably 98, 99% of the viruses are actually dead. If I just go somewhere here in the middle, 10 to the power negative 5.5, something like that, it's probably around 90%. But see that at the 10 to the power 6 dilution, that actually is the LD50 value. That means that the virus that you actually have, your dilution of the viruses actually have an LD50 of 50% at the 10 to the power negative 6 dilution. Now, it gives you a very interesting idea. This particular dilution of the virus, the LD50 value of that virus will actually vary in a huge amount. Just as we talked about that, think about that in the 10 to the power negative 6 dilution of the virus, half the virus particles are infective for a Semmeliki-Forest virus or SFB that we talked about. On the other hand, think about that, in the 10 to the power 6 viral dilution, only 1 viral particle is infectious for papillomaviruses. So what you will in reality will see that this is the measurement of your plaque forming unit, not the total amount of viral particle present in there. Because if you actually consider the total number of viral particle, this number will be very, very different for many different viruses. So we always actually measure the viral infectivity by the plaque forming infectivity. That means that in your sample, how many viruses are there capable of infecting the host. So that's why we always use this word, the plaque forming unit, not the total number of a virion in a sample. So the next part that we are talking about is the viroids. And these are the infectious agents which are composed of closed circular single-stranded RNAs. And they actually do not code for any gene products. And the replication requires the host cell, and the host cell DNA-dependent RNA polymerases will actually replicate these. This is an interesting idea, that they are RNAs, but they can be detected by the host or the plant DNA-dependent RNA polymerase to replicate themselves and will cause the plant disease. So here you are looking at two of the different types. So the top one right here, this structure that you're seeing, this is actually called a Apospaviroid, viroidae. This is the family. On right here, this is the one that you're looking at. This is called the Avsunaviroidae family. So these two actually vary in a structure. You can see that you have interspaced different regions where you can see that they're in this area P, T, these are the areas which are actually responsible for most of the pathogenicity for this viroids. And on the other hand, you can see that here the structure is different, and it is just a closed circular RNA, and they are extremely small and doesn't have any capsid surrounding them. So this is just a comparison that how small are the viroids. This is an E. coli DNA of the bacteriophage T2, and this is a human papillomavirus. You can see that HPV, a very, very small DNA right here. RNA of bacteriophage F2, S-shaped. If you just compare their tight size, this is the size of a bacteriophage. On the other hand, you can see, again, remember that this whole DNA of the bacteriophage can be packaged inside because supercoiling allows that to pack this much of DNA to be packaged inside. But compared to that, just look at this viroid, how small it is. Satellites, on the other hand, are infectious nucleic acid, either DNA and RNA, and the satellite viruses encode their own capsid proteins when helped by a helper virus. And satellite RNA DNAs do not encode their own capsid proteins. On the other hand, always a helper virus actually is needed for the replication of the satellites, always. And it encodes one or more gene products actually. Not only they require the helper virus for the replication, these satellites actually can enhance the severity of the diseases in some cases. Say for an example, so hepatitis D virus is not a regular infectious virus that you're found separately. When you have a hepatitis B virus infection, a hepatitis D virus is found as a satellite, not always. But usually it has been seen that if you find the satellite virus, it actually causes a much more severe hepatitis B virus infection rather than not having the satellite. Does not mean the hepatitis B virus infections will always have a satellite, but sometimes they are co-infected by these satellite viruses. So the last part that we are looking at is the prions. This is not anything to do with nucleic acid. So we have seen the viruses are actually nucleic acid, either DNA or RNA, which are enwrapped into a nucleic capsid. On the other hand, what we are actually going to see, the prions are completely composed of proteins. And this causes a variety of neurodegenerative disease in humans and animals. And it was first observed in the sheep where they lose the coordination. And this is actually known as a scrapie. Remember, that's scrapie is the disease. In the cow, it causes the bovine spongiform encephalopathy, because what happens that in the brain of these cows, which were actually seen after the infection, they form plaques and empty area, and this becomes a porous brain. And that is called the bovine spongiform encephalopathy, which is commonly known as a mad cow disease, because they also lose coordination due to this. There are several different types of disease in humans. This is also called a kuru. Here's an interesting story of kuru. Kuru was actually found in the indigenous people of Papua and New Guinea. And these 4 people in the Papua and New Guinea, they were actually cannibals. And the kuru was actually very common and reported in there. But the cause was not known until the 80s and 90s. and that actually goes to the prion disease. So we are going to talk about that in a second, that how this actually affected the Kurus. So what happened, that this is actually, the disease is caused by a protein which actually caused this neurodegenerative disease, and the protein cannot be easily destroyed. So cooking will not easily destroy these proteins. So what happens, that sometimes some of these cannibalistic people, they actually suffer due to the mental disorder, and that they will go crazy. And some of these people will actually think that they are under the influence of some kind of evil spirit or something, and they will actually kill them, and then they will actually burn and eat their meat. So this person who is infected with this infectious prion protein, all the people who ate that meat of that killed person in a cannibalistic way will get the Kuru. And this is actually an amazing story, that how this Kuru is due to this prion disease. The fatal familial insomnia is also another disease. It's called FF insomnia. This is also a neurodegenerative disease which is caused by the prion. Creutzfeldt-Jakob disease, which is actually called CJD. Then this is a big name. It's called Gerstmann-Straussler-Scheinker syndrome, or GSS. These are all different discoverers' name, but all of them actually affect one specific part of the brain, and the outcome is different. So it is all actually a neurodegenerative disease, but it just depends, the outcome just only depends that how they are infecting. So let's take a look at how this actually happened. So in every cell, so you can see that right here, this is a neuron cell. In every neuron cell, you have a protein which is called PRPC. This is a prion protein. C is for cellular. PR protein C. Each and every of neuron is present in the normal form. Now, this protein actually, while being manufactured in the cell by the translational process, after translation, the protein needs to fold into its three-dimensional structure. Sometimes this protein actually folds in a different way, which forms a PRPSC, or prion protein. If you remember that, we saw this prion protein scrapie right here. This SC comes from the scrapie. What we mean by that the prion protein SC is the infectious form of the protein. It changes its conformation to the abnormal form. Now, this is the interesting part. This protein, newly produced PRPSC, or the scrapie molecules, convert more normal molecules to the abnormal form through an unknown mechanism. And this is the sad part, that your normal PRPC proteins, if they are contacted by a PRPSC, they are going to convert into the SC form. So what happens, that if somehow a human being actually consumes the meat from where these prion proteins actually gets into the intestinal wall, from there they goes into the lymphoid tissues, then they make their way into the neurons, and from there, when in the neurons they reach the brain, these proteins in the neuron, if they actually touch another protein which is PRPC, they will convert them into PRPSC, and it will just go on, and that will kill the cell. So the prion protein scrapie will kill the neuron. So remember that the neuron cannot be replaced because they are amitotic, so they don't divide. So once a neuron is killed, that means its loss is permanent. So that's how that when you kill these neurons, it will form a plaque. And when these cells are very important into a particular area of the brain, they will actually cause these several different diseases. So this is how this is just an infectious protein, and that caused this disease. The sad part is that the prion protein cannot be even destroyed by just normal autoclaving, which can destroy the normal endospores of the bacteria, but not the prion protein. You probably need high alkali, like almost 0.5 normal sodium hydroxide. And then if you just actually autoclave in presence of those, it can actually cause that. No proteases can take care of this. And that's the reason that there is no drug for these. A person who is infected with this prion protein is actually one point of time is destined for death. There is no cure, not whatsoever. There is no medication. So the material is a complement of the chapters of the three domains of life. You can see that viroids actually can infect all the domains of life, archaea, bacteria, eukarya, and understanding that each member of the acellular entities is how they function, how they replicate, can round out a greater understanding of the microbial world. And the virology is an integral part of the microbiology. So that will be the end of your chapter 6.